Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm excited to be here today with Dr. Luis Diaz, who is the head of the Division of Solid Tumor Oncology at Memorial Sloan Kettering. He's also a White House appointee for the National Cancer Advisory Board, and he's a member of the National Academy of Medicine. I'm going to just very quickly go through some highlights of his career, but at a very high level, he's done pioneering work in precision oncology, really bridging the clinical application of novel cancer genomics, in particular developing targeted therapies for patients with solid tumors, and also some groundbreaking advancements in immunotherapy and really surveillance of cancer care using liquid biopsies and other kinds of novel methods. So Dr. Diaz, I'm very excited to have you with us here today and hopefully cover a lot of ground, including some very exciting data that you've presented recently.
Yeah, thanks, Patrick. I'm really excited to go forward with this conversation. I think we'll be able to get through some interesting stuff.
As I alluded to in the intro, you recently led, honestly, a groundbreaking trial that caught my eye when I saw the data, because you don't see this very often, which is 100% response rate to anything in this world, let alone cancer. I wonder if you could talk about that work, just for people who aren't familiar with it. But then I'd love to just rewind back actually to the origins of it. When did you start doing work in checkpoint inhibitors, immunotherapy, and maybe help us follow the thread to the breakthrough that you had recently?
Yeah, no, I think that study was a lot of fun, but it's really the culmination of decades of work. And to summarize, what we found there was that in patients with a genetic signature called mismatch deficiency, they are exquisitely sensitive to checkpoint blockade, immune checkpoint blockade. And when we pointed that concept, the concept of mismatch deficiency, which results on a tumor intrinsic phenotype that's highly immunogenic, and the existence of immune checkpoint blockade, to a scenario that's quite brutal, which is early stage rectal cancer that requires chemotherapy, radiation, and resection of the rectum, where a large portion of the patients require an ostomy bag, has major life-altering consequences, renders many infertile, and in terms of the self-esteem going forward, it can be quite devastating. Adaptable, but devastating, but the patients are cured. So the question is, in this marriage between cancer genetics and the immune system, could we see similar results that we are seeing in patients with advanced disease and early stage disease? So we designed this study to take a handful of patients with these mismatch deficient rectal cancers that traditionally get chemotherapy, radiation, and then surgical resection. And we treat them with immunotherapy, 6 months of immunotherapy. And we said, okay, at the end of the 6 months, we're going to assess them. If there's any tumor left, they're going to go down the traditional route.
Yeah.
But my colleague, Andrea Sursek, inserted in there, if by chance all the tumor is gone, let's proceed with watch and wait.
Right. Keep an eye on him.
Just keep an eye on them. Let's not do chemo. Let's not do surgery. Let's not do radiation. And that's what we did with the first patient. Tumor disappeared. No chemotherapy, no radiation, no surgery. And to our surprise, not only did the following 13 patients all respond completely, but they didn't have any residual tumor. And to date, all of them have been doing fantastic.
Oh.
We're up to 100 patients now. And in the rectal cancer cases, they're all behaving the same. They're melting away with no residual tumor. But we've expanded it to other tumor types that are mismatch deficient, because the biology of mismatch deficiency is that it may be more common in colon cancer, but it's also found in endometrial cancer, prostate cancer, bladder cancer, pancreatic cancer, gallbladder cancer, stomach cancer, esophagus cancer. And if you look at virtually every tumor type, The fraction might be small, but you'll always find a case or two of mismatch rare deficiency. And what we find is very similar results, that these tumors in other locations of the body also melt away and eliminate the need for traditional therapies like surgery. And we're most interested in tumors that are where the outcome of surgery can be devastating. The rectum was one, but you can imagine removal of the bladder, no bueno. That's brutal.
Yeah.
Removal of your pancreas, of your gall system or your bile system, your stomach, your esophagus. Those are devastating surgeries. Now they're life saving, and people can adapt, and they have adapted and lived great lives. But we wanted to try to avoid that. So that's the clinical result.
Yes.
But where did the story really begin? And it really began in the investigation of hereditary cancers, and specifically colon cancer. Probably more than 150 years ago, there was a investigator at the University of Michigan who identified these families who had a dense family history of colorectal cancer. And there was an investigator out of Nebraska, Henry Lynch, who categorized these patients and defined what we currently know as Lynch syndrome, a hereditary form of colorectal cancer that was associated with some other cancers like small intestinal cancer and so forth. And then if you accelerate from the early 1900s to the early 1990s, really it was a work from Bert Vogelstein, Albert de la Chapelle, and a number of investigators around the world who had very interesting cohorts that identified a replication error dominant subtype in these hereditary colon cancers. And what they found was that these tumors had replication errors in areas across the genome of repellent elements called microsatellites. And these errors were insertions and deletions that at the time were thought to be 6 to 20 base pairs, maybe even 50 base pairs in length, and that those would result in this MIN phenotype, a microsatellite unstable tumor. And along those times, there were a couple of other observations that were made or actually cataloged because they were known anecdotally for many years.
Right.
The first one was that these tumors that were hereditary had a dense lymphocytic infiltrate. This was published by Stan Hamilton at Hopkins and now at MD Anderson.
And so much more dense than other tumors, I guess.
Much more dense than most other tumors. It was very, and to the point where a pathologist would see it and say, I bet you this is a hereditary colon cancer.
Right. Interesting.
For decades and decades. So this was anecdotal. The second observation here was that these changes were happening in these microsatellites, and they would link to these cases that had the familial linkage. And then a few years later, again, out of Bert Vogelstein's group, they found the mutations that were associated with this microsatellite instability, and they were in genes that maintained mismatch repair. Now, this was guided by work done in bacteria, which received a Nobel Prize a few years ago in replication error defects and mismatch repair defect correction in bacteria, and understanding the mechanism around that. So in Bert Vogelstein's lab, they found the human homologs, and they were able to catalog mismatch repair deficiency in a large way. The other observation done around that time was that these microsatellite unstable tumors were abundant with changes across all the microsatellites, which numbered in the thousands, and also didn't repair mismatches across the genome. So they were hypermutable. So pause there. That identified these hereditary syndromes. Then when they looked at the non-hereditary cases, a fraction of those were also microsatellite unstable. So you had microsatellite instability or mismatch repair deficiency in a subset of colon cancers, a fraction that were hereditary, and a fraction that were spontaneous or not hereditary. And so the sequelae of mismatch repair deficiency that occurred early during tumorigenesis, not at its origin, but probably soon thereafter, regardless if it was hereditary or not, led to microsatellite instability, so multiple insertion deletions of microsatellite that can be measured, and an exceptionally high tumor mutational burden. And as a corollary to that, they also had a dense lymphocytic infiltrate. That work probably ended in 1994. And following that, it was just implementation of that. This might just define a hereditary subtype.
Right.
There was no clinical sort of step forward.
Right. So what do we do with this clinically? Yeah.
And so there was also an understanding that these mismatch-deficient tumors were more resistant to chemotherapy. Why, we don't know. My hypothesis is that tumor heterogeneity leads to resistance to most therapies.
Yeah.
Because you have a pre-existing clone that will resist selection by that therapy. In a similar manner, these hypermutable tumors have such a heterogeneous pool of cells that one's going to be resistant. So therapeutically, not much happened to this population of tumors. And then in 2010, 2011, our lab was responsible at the time for being involved in the genomic sequencing of cancers for the first time. And Bert Vogelstein at that time was publishing sequences of cancers using Sanger sequencing, if you can believe it or not.
Yeah.
Which is crazy because it's very manual. And one thing became very clear is that the landscape of mutational burden across tumors was different. So pediatric tumors, sporadic tumors had a relatively low number of mutations per genome. A sporadic tumor like breast cancer or colon cancer that was not mismatch deficient could have 75 to 100 mutations per coding exome. And then there were the mutagen associated tumors like melanoma or lung cancer that had between 150 and 300 mutations per coding exome. And then you had the mismatch deficient tumors that had 1,000 to 3,000 mutations per coding exome. Big difference. And that was an observation that was made. And at this time, immune checkpoints were also coming to the forefront. And immune checkpoints, starting with CTLA-4 and then eventually with PD-1 blockade, were showing fantastic responses in melanoma and kidney cancer. Not surprising. These are immunogenic tumors. You can treat them with IL-2. Many respond.
Right.
In fact, some are cured. And no one really knew why. We just chalked it up. They're immunogenic for some reason.
Right.
But what really caught my attention and the world's attention was that lung cancer was responding. And one idea that we had in our group was that maybe it's the amount of mutations. And could these mutations, if transcribed and translated into proteins, be seen as foreign to the immune system? And therefore, if you had many, like thousands, the chances would be higher that they're immunogenic. And
it's like a billboard for the immune system, basically screaming out.
A little bit. And so we thought then the most immunogenic should be these mismatch deficient tumors.
Right.
And so at that time, all the companies who had these drugs were going for registration, particularly in melanoma and in lung cancer, because those were the ones where they were seeing signals. And we had seen a case of colon cancers that were treated with anti-PD-1 therapy. And there was one case that responded. And we actually asked the investigators, because they had no idea.
Right.
Check it for mismatch deficiency. And lo and behold, it was mismatch deficient. And so we went to multiple drug companies to try to get checkpoint inhibitors for this concept. And they all said no, with the exception of Merck. They wanted their registration of the drug unvarnished.
Sure.
For these big indications like lung cancer and melanoma.
Yeah.
Things where it made sense. And so Merck said yes, their drug was still investigational, but they said, listen, you have to pay for the study. You have to hold your own IND, and we'll let you cross-reference our safety package. So we had a drug. We had what we thought was a good hypothesis. High mutational burden with immune checkpoint inhibition would lead to responses, but we didn't have money. And so at that time, I had begun a relationship with a group called Swim Across America. And they were based nationally, but we had a swim in Baltimore where 500 swimmers would jump into the Chesapeake Bay and raise money for cancer. And they decided to fund this.
Wow.
And with $500,000, we began the clinical trial. And because this was investigational, we couldn't start it with freshly diagnosed colon cancer that was mismatch deficient.
Yeah.
We had to start with metastatic cases, and we had to treat patients who had failed all lines of therapy. So we're getting pretty sick patients.
Yeah.
And what was remarkable is as we started treating patients, in many cases, tumors started melting away.
Wow.
These were patients that were bound for hospice or in hospice. These were
Wow.
patients who were having severe pain. And what we saw was in the first 25 patients, 60% of them had their tumors virtually go away.
When was that? How many years ago was it that you
That
started
was
seeing
we
that
were, we
setting?
were seeing this 2011, 2012,
Wow. It's going back a decade.
2013.
Yeah.
And we started the study around 2012, 2013. And then we were seeing the colon cancers that were mismatch proficient, so low number of mutations, they weren't responding. The tumors were growing right through this. So in 2015, when we were ready to report this, two papers came out from Memorial Sloan Kettering, actually, that were very interested out of Tim Chan's group. He had sequenced tumors that were treated with checkpoint inhibition with melanoma and showed that the ones with more mutations responded better than the ones with low, retrospectively. And then in the lung cancer cases treated with anti-PD-1, he showed that tumors with more mutations did better than those with low mutations. So giving some support to what we were hypothesizing in the study we were doing prospectively. So when we reported in 2015, it was a pretty big deal. We found a mutational signature that in patients with metastatic disease that were treatment refractory, that we were controlling the tumors in ways that we'd never seen with just immunotherapy alone. So that was in colon cancer. That same study had a bucket of non-colon cancers that were mismatch deficient from any tumor type. And we looked at that and they were responding just the same way that tumors that were mismatch in colon cancer. They were about 60% were having major responses. And we published that in 2017. Soon thereafter, the FDA gave us breakthrough status, and then subsequently in 2020 approved checkpoint inhibition for tumors of any site with mismatch deficiency could be treated. So it was the first FDA clearance of a tumor agnostic indication.
Wow.
And so that really kind of opened the floodgates to tumor agnostic approvals going forward. And so the story could have ended there, right? There was some housekeeping to do. Could you bring this up to first line site patients? And that's where we stood. But we've since then described the study that you led off with, which was the rectal cancer study, where we brought it to before surgery. And we're also bringing this to after surgery. But last year, we published a paper that probably didn't catch the media or the scientific community's attention as much as it should have. And it's the things to come, which is, could we use this approach to prevent cancer in patients who don't have cancer?
Yeah.
And we looked at a population of patients with Lynch syndrome. They get cancers at a rate of about 4% per year. That's a lot.
Yeah.
Take
There's
100 Lynch syndrome patients in a room per year, and 4 of them will get cancer. And we had 2 questions. In the colon cancer subgroup, we know that the evolution is normal tissue to polyp to cancer. And we know that in a large majority of these, they would develop mismatch deficient MSI high tumors.
Yeah. What percentage roughly would it be MMR deficient?
In the high 90s.
Right. Okay. Wow. Yeah.
But a fraction of them wouldn't be. They would just be get cancer like everyone else does. But could we prevent polyp formation in these Lynch syndrome patients? And that was the first question, and that was super interesting to us. And then the second question is, could we prevent cancer formation or delay it?
Yeah.
And so what do you think? Do you think we could prevent polyp formation with PD-1 blockade in these Lynch syndrome patients?
My guess is no, because I'm thinking the fact that you shared earlier about the kind of immune system swarming on the tumor doesn't happen quite that early, but I'm hoping to be educated if I'm wrong about that.
So you're right. So we saw no different in polyp formation. And the question was why? You know, these are Lynch syndrome patients. Maybe the tumor is the second hit because they have one inactivated allele
Yeah.
of their mismatch repair protein, the second hit, and then they take off the cancer. And that's not how it happens. Most polyps in Lynch syndrome patients are mismatch repair proficient. They get
Okay.
the second hit down the road. The proliferative neoplasm, which is not malignant yet, the polyp, that is dividing, and that division allows for a second hit to happen. So the
Interesting.
polyp isn't mismatch repair deficient. They
Hmm.
become mismatch repair deficient tumors because they have a propensity for that,
Right.
but they're not. The polyps are not that. So we weren't
Why,
seeing.
why, why do the polyps start to show up earlier? They're not.
Ah,
What do we know
so
about that?
so we think that mismatch repair deficiency may lead to a a propensity to develop polyps. And there might be a hypomorphic second allele that leads to some sort of
Yeah.
fitness advantage.
Interesting.
At the very least, a more permissive environment for mutations to be accumulated.
Yeah.
It's not a higher rate of mutations, although net on net it would be. It's that you have your basal background mutational rate at approximately one mutation per 10,000 bases, and you accumulate those over time.
Right.
So if you knock out mismatch repair on day one, your cell's not MSI day two. It's not hypermutant on day two.
Yeah.
It's not immunogenic at day two. In culture, you know how long it takes? A year.
No clue. Oh, wow.
And so in
In culture.
humans,
Yeah.
in
So in humans, it's
culture,
going to be much longer.
it's much longer. And so you're not immunogenic or MSI or hypermute or high tumor mutational burden until 9 months, 10 months, 11 months, and then you become immunogenic.
Interesting.
And so that explained it to us for polyps. So we were a little disappointed because we wanted to see, can we eliminate the polyp?
Right.
But what about the cancer? And then patients who were high risk. And what's high risk? Someone who already had a cancer. If they got anti-PD-1, you could reduce or delay the onset of cancer by about 80%. So there is some impact there. And so I think that opens up the possibility for immunoprevention in a way that hasn't been possible before. So that's number one. Number two, this was both exciting and unexciting. When we looked at chemotherapy in these patients that were high risk, we also reduced it about by 70%.
Oh, interesting.
So not just immunoprevention, but I think there's a gap and an opening for cancer prevention as well. With
Is checkpoint inhibition generally, how are the symptoms, side effects compared to
yeah.
chemo?
So in advanced patients, we do see a fraction of side effects. Typically, you're turning on the immune system, like autoimmune
Yeah.
diseases or certain tissues. In the earlier stage patients, we've seen less. So my hope is that that's something that will continue.
Yeah, interesting. You've more broadly, you've done a lot of work in liquid biopsy that I think fits in this
Yeah.
general paradigm of prevention. Maybe you can talk about that and
Yeah.
where I'm interested to hear where the state of the art is right now and where we might get to because it's it's the holy grail for the field to be able to do this, right?
Yeah, I think what I missed in the story is that when I was a fellow in Bert Vogelstein's lab and trained with Bert Vogelstein, Ken Kinsler, and was contemporary there with Victor Valculescu and Nick Papadopoulos and Shibin Zhao, was that I joined a cancer genetics lab because I didn't think the immune system would ever help solid tumors.
Right.
And so my mandate in that lab was, can we figure out how to use any of this cancer genetics therapeutically? And I'm not a chemist, and my training wasn't necessarily to make drugs that would target these mutations. So the areas I was drawn to were, could we use these mutations as markers of prognosis or predictive markers for response? But there was work going on in the laboratory that predated my arrival in the laboratory using a technique called digital PCR. And that was really pioneered by Bert Vogelstein and Ken Kinsler, where they took PCR, which was semi quantitative, where you could amplify amplicons and see bulk amplification rates, or over time with real time PCR, measure that difference using evolution and template density at the initiation of that PCR reaction. And so what Burt and Ken did was take those amplicons and reduce them to a single molecule by a dilution in a Poisson distribution, and they were able to count in a digital sort of way, positive and negative. And what that really did, it didn't make what we did fundamentally with PCR better, but it reduced the background. Because if you amplify anything to 40 cycles, you can get anything to amplify.
Right.
But if you can take it down to digital and you could titrate the number of cycles, you could get a very precise difference in positive versus negative in a digital way. And there you were counting positive versus negative well. So you made it digital. And that was really targeting an approach that Burt was pioneering, which is, can mutations in DNA be used as biomarkers for the detection of cancer for a variety of different tools? And he was focusing on stool DNA. And so in 2004, 2005, 2006, we began looking at that. Could you use that to look at rare fragments of DNA in the blood? And the idea had always been, can we use it in the blood for early detection? And given my background and training, that I was more interested in, can we use this to monitor things about advanced tumors? And so we began research initially in the mouse with a graduate student at the time, Carlo Rago, where we were looking at human line 1 sequences from human xenografts in mice just by a tail pinprick. We could get 1 drop of blood, 50 microliters, spin it down in a solution, an EDTA solution, extract the supernatant, and we could actually detect this line 1 human DNA in the mouse that were in short fragments, about 100, 200 base pair fragments, and show that we could amplify those, and that we could show that tumor burden, when the tumor grew, it would increase, and then when the tumor responded, it would decrease, and then when we cut out the tumor, it disappeared, and that the half-life of these tumors is very short. And so we then moved on to humans, and what we could detect there was, not only could we detect mutations that were present in the tumor in the blood, we could genotype the tumor by blood draw, but we could track the tumor growing and decreasing, and we determined that the half-life was only about 2 hours. And we determined that if patients underwent surgical resection, that if there was a residual circulating tumor DNA, because the half-life was so short after that surgery, a day out later or a week later, that meant that there was still tumor in the body,
Right.
and that the tumor would recur. And if there was no tumor left, that the tumor would likely be cured. And subsequent to that, we had papers that we published that showed that we could not only detect this in virtually any tumor type, but we could track minimal residual disease in a very precise way, and we could track resistance. So if we treated with an EGFR inhibitor, we could track the development of resistance over time to that,
right,
based on mutations. And so that whole world kind of opened up from those initial papers. And subsequent to that, not only were there scores of papers, I think, replicating what we did, but there were companies that formed, like Guardant, like Grail. Exact came out of a lot of work that came from Burt's work. And more recently, Natera, Haystack, companies that took on this technology, and companies that we found in personal genome diagnostics, Delphi, and so forth. And so what's been interesting is that whole ecosystem has evolved. And so from those simple experiments where we're looking at one mutation at a time, they've kind of evolved to looking at panels. And I think the next evolution is really doing whole genome in a way that is not only effective, but cost effective. And the applications have expanded. Minimal residual disease is a real thing. Tracking mutations and resistance mutations over time is a real thing. And I think early detection will be a real thing. And so I think that landscape has been impressive. And from a technological standpoint, the tools to identify mutations that are bona fide mutations from artifact using next generation sequencing have evolved. And that approach has become much more sophisticated, not only by the quality of the sequencing, but understanding the steps before the analytic steps and the wet lab based steps, and applying tools to bioinformatically remove artifactual mutations from bona fide mutations. Duplex sequencing, so the 5' strand mutation has to match the 3' prime mutation in the template DNA, I think is a big advance that we'll see.
Who does that? Who's doing that? Is everybody doing that now? Or is that
Everyone's doing that now. Probably the first examples of this doing for liquid biopsy were again out of Bert Bolstein's lab. Studies led by his group really showed the duplex sequencing. There are other groups that have done this as well. The Loeb group out of Seattle had some work around this, but most of the groups are not doing that. There is now a sequencer, an Ultima sequencer that has this baked into its
interesting.
It's process where it's doing PCR-free emulsion library production so that you get both strands and you get duplex reads. And I think that's one of the advantages of their what's called their PPM-seq. And so I think those are steps forward.
Yeah. You laid out this kind of tale of two cities in some ways where there's a ton of value from using this kind of testing to guide treatment, understand evolution. People have been fairly negative on GRAIL and some of the others that are doing really early detection. I'm curious whether that negativity is warranted in your view, and if it is, like, what's missing there? Is it a technology limitation? Is there there's a fundamental challenge with sensitivity of screening in healthy or near healthy people, right? I'd love if you could just talk a little bit about what it takes to get to that early detection that is just hard. It's very hard and nobody seems to have cracked it yet.
I'll keep talking a little bit about the evolution of the technology. So now teams have been looking at fragment size because tumors have smaller fragment sizes. There's been a lot of work at looking at the end reads of those fragments. And there is fascinating work out of Dennis Lowe's lab, who was one of the pioneers in cell-free DNA for non-invasive prenatal testing, looking at long fragments. Cell-free DNA is always considered to be short fragments. There's also long fragments in circulation. And I think that's quite interesting. And then the adaptation of machine learning to large data sets to that. And so all that's what that is giving us from a technical performance standpoint is reducing background and amplifying signal.
Yeah.
And the signal we want is cancer, right? Over background. And because cancer is so similar to self, that delta, the more we can amplify that delta, the better. Laboratory-based methodology probably gives you the best performance that's applicable in any scenario. Machine learning-based techniques tend to perform quite well, but are subject to batch effect. So, but let's leave that to the technical performance of the assays. And now let's move the attention to biological performance. What's ground truth?
Yeah.
Right. And so ground truth is that a tumor cell that's in the body, by some mechanism, we believe it's apoptosis, releases DNA, both bound to nucleosomes and free DNA not bound to nucleosomes. And big tumors release a lot.
Yeah.
Small tumors release little. One cell releases very little.
Yeah.
Into a pool of interstitial fluid and 5 liters of circulating blood fluid in an adult.
Right.
So the likelihood you'll find one fragment from one cell dying is probably fairly low, right? Just by chance.
Yeah.
So that's the biological limitation, right? So that's where whole genome sequencing or large panels become more possible, because you're just not looking for one allele of, let's say, a KRAS gene. You're looking for multiple mutational events.
Yeah.
And so that increases your chance a bit. And so I think that where approaches, actually, I'll pause for 1 second. So let's go back to the technical part. There are other approaches, methylation, that see many fragments altered. And the methylation signal there is broader because it's not just tumor specific, it may be field effect specific. So you might have a 1 centimeter lung tumor, but the whole area that's affected by smoking or some environmental pollution is altered. So you're defining a higher sensitivity. So if we move to things like GRAIL, they're looking at a methylation biomarker. Their sensitivity is a little higher, but their specificity is not that great. So about half of the tumors they detect are potentially not tumors, or we can't find the tumors. And so that's the challenge there. Exact inherited a technique from a company I was involved in. It's called Thrive, that was purchased by Exact, that looks at mutations. They're adapting that. Again, slightly lower sensitivity, but a much higher specificity. And then you have companies like Delphi that are looking at fragmentomic-based early detection, and they have a very high sensitivity, but their specificity is lower. They're able to do that to enrich for a gold standard test like lung cancer screening.
Right.
So to sum it up, I think performance is still mediocre. From an individual standpoint, it's probably better than not having a screening test. But from a population standpoint, it probably is too expensive.
Yeah.
And it will lead to a lot of anxiety, unnecessary medical tests. But do you remember the cell phones before Nokia? These big, like lumbering cell phones?
I'm aware of them. I never owned one, but I have seen them.
Imagine what we have now.
Yeah.
So we're in the Nokia phase.
Yeah. Is it an engineering problem in the sense that actually one or more of these techniques, either alone or together, could be combined to crack it? Or is it a scientific problem? Because you described this like quite challenging signal to noise ratio. You've got a tiny tumor releasing into 5 liters of blood. Like, is there a marker that we haven't discovered yet that could pull that signal to noise out? Or is it like if you push the fragment length and the methylation to an extreme enough degree, you could get that resolution?
Well, let's say we start moving into like we want to fantasize about what's possible, right?
Yeah.
If you look at a KRAS allele, and that's one mutation in one allele, right? One mutation in 30 million base pairs, right? And that's diluted down by 60 million base pairs, right? So you have the fragment, you can align it to the KRAS, you can find the single base pair change. That's one signal per cell. Methylation probably amplifies that to probably by 10 to 100x, right? But it's less specific. Yeah. It's not the tumor cell, it's the tumor cell and its neighbors, right? Fragmentomics, I suspect, is going to be something similar. But let's say we went to RNA. That KRAS is probably 10 to 100 to 1,000x more amplified, right? But RNA is not stable.
Right.
Unless it's bound to a protein or unless it's protected somehow, it's just not stable, right? And so I think there's a challenge there. I think with high abundance tumors, that might be possible, but not for early detection. But what's the next step after RNA? It's protein. Could you find a mutated protein? And I think that's going to be probably another 10 to 100x beyond RNA, maybe.
Yeah.
Depending on the stability of the protein. And so I think if we were able to engineer something to detect mutations that were expressed and survived in the circulation, that might be a higher abundance signal that we can detect earlier. But DNA is still a very stable template, and we are detecting 30 to 40% in its ideal state of stage 1 cancers. That could be probably increased. So that's one side of the problem. So it is engineering to some degree.
Is it fair to say that specificity is the more important part of it to crack, from the point you made about individual versus public health? Like, if the specificity isn't extremely high, then it just becomes a problem to all the additional screenings you end up paying for.
Well, it depends if you have something to fall back on. If you have an enrichment test that was, let's say it was you detected every single cancer,
yeah,
but half of them were wrong.
Yeah.
So obviously that's less than ideal. But what if they all still got colonoscopy?
Right.
Let's say the whole population needs colonoscopy at age 45, right? But you put this in front, it wouldn't miss a tumor, but it may allow you to exclude patients that don't need a colonoscopy then.
Yeah.
So
it's complicated.
You can scenario map it, right? And you can say the same thing for lung cancer, or cervical cancer or skin cancer, where we have bona fide gold standard screening techniques. For all the other tumor types, it's a problem.
Yeah, that
So
makes
that's
sense.
one side of the problem. The other side of the problem is, but you're right. So let's say you had a tumor, a biomarker that detected 5% of all tumors, but with perfect specificity, right?
Yeah. You take that. That's freebie, right? You
Yeah,
take
it's a
that all
freebie,
day.
but what's the issue? It's not a freebie. It's expensive,
Yeah.
right? So to deploy that, let's say just basic cogs, and let's say you, DNA extraction, maybe at scale it'll be under $5, maybe. Right now, not at scale in the lab, it's $20, right?
Yeah.
Okay, so let's say you do a next generation sequences approach and you do library prep. Maybe at scale, maybe it's going to be $50, maybe, right? And so, and then let's say you go to whole genome sequencing, that's going to be cheaper. You can get that at $50 at scale, maybe $20 at scale, right? Depending on how deep you go. And then you have your bioinformatic, and let's say you fully engineered it and it's just electricity.
Yeah.
That's going to cost you something. And then your FTE, right?
Yeah.
It's hard for me to imagine. And then overhead, right? Let's put $20 on overhead. Less than that. Let's put $5 on overhead. It's hard for me to imagine the cogs around that test being less than $100, 150.
Yeah.
And then we didn't even talk about shipment of the sample and the tube. So Streck or whatever tube you use still gets another at scale. Let's say $5, and shipment is still going to be, again, at scale maybe $10, $20.
Yeah.
And then phlebotomy, right? So
Yeah.
the whole thing,
and you got to do it every year, probably.
$100, right?
Yeah.
And then whoever makes it has to make some money, right?
Yeah.
And so it's
a tricky problem.
The cost is still there. Now, if you could get it down to the whole, just running the test to under $10, then the performance becomes a little less of a problem, because then you can apply it to a large population, and then the medical system will figure it out.
Yeah. Yeah. Yeah, it's interesting, and you get an incremental bump in sensitivity, specificity, but and the system figures out. Yeah, it's an interesting way to think about it.
Yeah. Now, if you have a home run test that's super highly specific and adequately sensitive, then I think you have something real.
Yeah. Wrapping up here, I want to ask you a curveball question, which is what can other fields outside of cancer learn from everything that, the work that you've done, and more generally, because cancer, you led the first, FDA approved treatment for focusing on the genetic mutation in question rather than the tumor type. We're far from that in every other area of biology, and so many other things are, decades ahead in cancer. What lessons do you think we can bring across and say all these things will happen everywhere else, and what are things that actually are not the same, because cancer is just fundamentally different?
So I'd say, and this is a great question, because for a number of reasons, but one is, so liquid biopsies, can you apply them to autoimmune and inflammatory diseases? Can you apply them to heart disease? Can you apply them to lung and liver disease? Can you apply them to brain diseases? And I think that there's active research going on there.
Yeah.
It's really exciting. Same pathway. Can you track disease? Can you see response to therapies? It will help that whole ecosystem, because when you understand what response is, then you can properly design studies and track therapies.
Yeah.
And then eventually moving all of that to early detection. That's from a diagnosis part. From a therapy part, I think that one of the interesting points is that I think other diseases will be caused by mutations. And we know that from a hereditary standpoint, right?
Yeah.
That there are many other diseases that are caused by germline mutations. But I think somatic mutations will also be involved in other diseases. They just won't be proliferative diseases.
Yeah.
And I think that's an interesting. There's a whole entity called clonal hematopoiesis. There's a whole entity of other clonal mutations causing diseases like endometriosis or aneurysms. But I have a company right now in stealth mode that's really interesting, which is, can we take precision medicine to autoimmune disease? And we're working on autoimmune diseases where we understand the autoantigen.
Mm-hmm.
And is it the whole antigen, which is typically a large protein, that causes that's causing the disease? Or is it really a precise area, almost like a mutation?
Right.
And we've defined the precise area that is pathogenic for many of these autoimmune diseases. And we've developed a therapy that can immunoabsorb, let's say it's an antibody-based autoimmune disease, that just antibodies that bind that one area, and you can neutralize disease. And I think what's exciting there is that you're bringing the principles of precision medicine from cancer to autoimmune disease slightly in reverse by defining the exact location of the genetic alteration or the immunological lesion that is being attacked.
Interesting. Very exciting. If people want to keep up with you, keep up with your work, they can obviously check out your website on mskcc.org. Is there anywhere else conferences you like to hang out? Should people follow you on LinkedIn? What's the best way to keep track of your work?
I think LinkedIn is a good one, but in general, AACR, ASCO are good meetings for us on the cancer landscape. But we're so interconnected now that if something big comes out, hopefully it'll be noticed.
Absolutely. Well, thank you. This was amazing. I learned a ton from you, and congratulations on all the amazing work you're doing. It's incredible.
Thank you.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selective Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.